All right, gonna make a quick video for essay number 3 of unit 3. So in this one, we're actually gonna go over blood. We're gonna go over the components of it. So kind of what makes up our blood. And then I'm gonna talk about plasma and some characteristics that we need to know about plasma briefly. Then I'm gonna talk about one of those components in more detail, erythrocytes, red blood cells. And we're going to talk about erythropoiesis, which is the formation of red blood cells, and some of the things that we need in our diet to make those as components for red blood cells. Then we're going to talk about hemolysis, which is the breakdown of red blood cells, because we know that they last about 150 days, and so we're always recycling them. And then I'll talk about hemostasis, which is actually kind of being in homeostasis with your blood. As blood cells are going away, we replenish them, and so hemostasis is actually the process of, if you got an injury, we're able to clot that pore, that opening with a scab, and we're able to prevent increased loss of blood. So this is kind of a lot, but we'll go through it as quick as we can, okay? So the first thing I want to talk about are the components of blood. Okay, I like to draw it in like a vial. If we were to take a sample of blood, okay, we would have kind of 3 tiers here. We would have our white blood cells, we would have our red blood cells, erythrocytes, leukocytes, erythrocytes, and we would have our plasma. In this, we also have platelets. Okay, plasma is about 55% of your blood. White blood cells are usually when you're sick. They fight inflammation. High amounts of white blood cells means cancer, leukemia. Okay, and then red blood cells, these are called our erythrocytes. And red blood cells, high testosterone, leads to increase in red blood cells. Right. Something in plasma, we have Na, sodium, we have calcium, or Cl chloride, we have potassium and magnesium. Okay, so those are some interesting and important components that we would have in our blood. Okay, so platelets, I believe, are going to be kind of part of the plasma because we have colloids. There are colloids in here, so albumin. We also have fibrinogen, which is clotting, dealing with the clotting cascade that we'll talk about with hemostasis. And then we have globulin, which deals with antibiotics. And antibodies. Okay. So these are kind of the components of blood and some of the things that we kind of want to know about it. So let's talk about the formation of a red blood cell. erythropoiesis. Okay. There's some things that we kind of want to know about this, and that is this is taking place in the bone marrow. Okay. And then we kind of want to talk about some things in our diet that kind of play a role in components for this process. So hopefully we can see that, yeah. Okay, so something that we want to do is we want to talk about some components that get into our diet. So we have our stomach here, and it leads into the duodenum. Okay, and so sometimes we can eat folic acid, B12. There's one other one that we kind of want to get into our diet here as components, and that's iron, Fe2+. And then we also kind of want like fatty acids. We want proteins, and especially carbs, because they'll be building blocks for these erythrocytes, okay? So, how I like to explain this is, we take in these components here into our diet, as building blocks, and then we put those into the duodenum, and we kind of start going into our diet. You know, we start digesting this and using it, right? What ends up happening is, if I were to take a cross section of this, and we had this kind of blown up here as a cell, what we would find is these components of iron, and then our folic acid, right, and then our B12. B12 uses a special factor with it called an intrinsic factor to help it be digested. So intrinsic factor. Okay, B12 intrinsic factor. And so one thing about iron is when it comes through the cell and gets absorbed, it actually turns into iron 3+. Folic acid will continue as folic acid, and then B12 comes through because of the intrinsic factor. And then we'll show different components or places that they go to up here in the cell. So then we have hemocytoblasts that occur in the red or in the bone marrow, right? And what these guys do in the bone marrow is they end up turning into myeloid stem cells. Okay, and from myeloid stem cells, they then convert into the three components that we're interested in: platelets, red blood cells and white blood cells. We're most interested in erythropoiesis in red blood cells and the production and development of those. So, iron plays a big role in part of this production. And these myeloid stem cells, let's see. Okay, I forgot one thing. You have the kidney as well. And the kidney makes a product called EPO. Erythropoiesis. And EPO, if there is low oxygen, then this EPO, in this case, EPO can secrete to the bone marrow area, and we have EPO, okay? And it helps with the production and, well, it actually goes to HGF first. And then it'll go to EPO. Excuse me. And then we also have something really important happening here with so this would be EPO. So then vitamin B12 and let's see, we forgot one. We have carbs, fats, proteins. And what ends up happening is these guys here will also secrete through the cell. But what they do is well, I think it's folic acid actually is what I'm trying to say is vitamin B12 and folic acid, they actually come in and they're a part of the DNA maturation step for these red blood cells. Then the iron and the carbs and the fats, they actually play a role in the heme production. Okay. So iron, carbs and fats, they play a role in heme production, which will make sense when we talk about hemolysis and the breakdown of red blood cells. So here we have heme production that's going to take place. And so that's why these components, and when we digest them and then we use them and facilitate them through the duodenum and then use them as building blocks with EPO from the kidney, we can actually build these red blood cells. And so if I were to scoot this down a little bit like this, and so what we end up seeing that happens is right here we have, our bloodstream. Can you see that? Bloodstream? Yeah. So here we have our bloodstream, and so these red blood cells can actually come into the bloodstream. And they come in as these immature red blood cells. And one thing that we want to know about these red blood cells is they have no mitochondria. have no DNA. Well, they have DNA, but no nucleus. But they have no mitochondria. Okay? So that's important to know. So then we get into the bloodstream, and these red blood cells, they come over and they are immature. And so we call them reticulocytes. Right? And then we have them mature into a red blood cell. Okay, and we call these erythrocytes. Okay? So then we have mature red blood cells, erythrocytes, and so they carry the gases, and then they actually have a short lifespan. So they carry O2 and CO2 for the exchange of the capillary beds, these red blood cells, and then they also do, they only live about 120 days. Okay? So if they only live for 120 days, then we got to think, okay, what causes these to then break down? But before I get there, I want to touch on this one more time. Here we have our blood components. We have platelets, or we have plasma, red blood cells, and white blood cells. White blood cells are going to be your fighter cells. In the case of leukemia or cancer, high white blood cell count. Red blood cells, these are going to be carrying your O2, carry gases such as O2 and CO2, okay? And then the other thing that they do is they live 120 days, okay? And then we talk about plasma, and we know that there's 55% of the blood is plasma. Fibrinogen is clotting agent in plasma with platelets. We also have colloids and albumin that make up the plasma. And then we have globulins, which globulins are going to be for antibodies and antibiotics and things like that. So we have globulins within this plasma liquid. And then we have the red blood cells like I was talking about. So if you have an increase in testosterone, you're going to have an increase in red blood cell count. So some other things that affect this is if you're at low elevation, O2 is low, so you increase red blood cells. I believe I have that right. Yeah. High elevation. Yeah, if you're at high elevation, then O2 is low. You can also have a hemorrhage. What a hemorrhage is a hemorrhage is going to be a loss of blood. We could be in hypoxia or in the hypoxic state, and this causes low O2, and so we increase red blood cells. Okay, and then anemia. Low blood cell count, and we would have a decrease in red blood cells because this decreases B12. If we don't have B12, we don't have DNA maturation as much, and we don't make as many red blood cells. That should be everything on here. Okay, so for this one, as far as this process goes, we have the formation of red blood cells, erythropoiesis. So what ends up happening with erythropoiesis is we end up having the development of red blood cells occur within the bone marrow. And we find that there are these basic components that attribute to building up these red blood cells, erythrocytes. And if we draw out the esophagus and the stomach and then into the duodenum, we find that we have iron that comes in, we have folic acid, we have B12, and then we have all of our basic macromolecules, our carbohydrates, fats, proteins, lipids, things like that, that will come in and be components of building blocks of red blood cells. So we end up coming in and we have a cross-section of the duodenum. We show how each of these get across. B12 is really important because it uses something called an intrinsic factor. And so what ends up happening is sometimes you can have anemia, and anemia leads to a inability to use the intrinsic factor, which then leads to us not being able to absorb B12. If we don't absorb B12, then we have an inhibition within the development process of these red blood cells. So what ends up happening is we have all these components, and then we have the kidney that also attributes to this with EPO. If oxygen is low, then EPO plays a role in building red blood cells. Because if oxygen is low, we need more red blood cells to carry as much oxygen as we can. So what ends up happening is EPO then gets activated, and then it activates HGF, which then allows EPO to leave and go to the bone marrow. In the bone marrow, we then have hemocytoblasts. They make myeloid stem cells and lympho cells, but we're not talking about lymphatic system, we're just talking about the blood. So we have myeloid stem cells, and then those will make the red blood cells, platelets and white blood cells. And so how do we control for them to make red blood cells? Well, these factors control that. TPO attributes to them making red blood cells. Folic acid and B12, they're gonna be a part of DNA maturation. All the macromolecules, carbohydrates, proteins, fats that I mentioned, those and iron, they help make heme for hemoglobin. So heme production. And so we have red blood cells, and then they leave the bone marrow and they come into the bloodstream as reticulocytes. And then they become mature red blood cells that last about 120 days, and they carry gases, okay? So that is the process of building up these red blood cells and erythropoiesis, okay? So now let's talk about erythrolysis. Or we call it hemolysis. Let me make sure I got everything now. Yeah, I think it looks all good. Okay, so for this, I'm gonna kind of draw some things out similar. So here we would have what's called a macrophage. Okay, and in the macrophage, we then have our red blood cell, and it's made out of two components, right? Blood cell protein, and then we have heme, and then we have our alpha and beta subunits from the globin part. So this breaks down to two alpha subunits, and this breaks down to two beta subunits. And then those both come together and they form amino acids. Okay, so the globin can be broken down into amino acids. The heme part gets broken down into the iron 3+. Which then produces erythropoietin, which then produces erythropoietin, and then erythropoietin will then produce ferritin. Okay, and we get these clusters. Then heme will then create biliverdin. And biliverdin will then go and create, through an enzyme, and this one is H2O soluble. But what ends up happening is we create bilirubin. And this bilirubin is not H2O soluble. And it's also unconjugated. And then this goes into our bloodstream. Okay. This goes into our bloodstream, and when it goes into the bloodstream, it exists in here as unconjugated bilirubin. Okay. Then we have something over here called the liver. And the liver will send up and recycle a product called albumin. And albumin will grab on to that unconjugated bilirubin and take it here. Then we add unconjugated bilirubin to glutaric acid, glucuronic acid, and the glucuronic acid, because they form together, it then kind of kicks it out of here, of the liver, as a conjugated bilirubin. So then, now it's H2O soluble. So now we have this bilirubin that's water soluble. And then this bilirubin then gets fed through the bile duct. And I don't think the bile duct really does anything, you know, because we have the bile duct with the pancreas. And then this bile duct will lead to the duodenum, and it goes through that sphincter of Oddi, right? And then in the duodenum, we then have this thing called protease. And protease is a bacteria, and it cleaves off sugars. And glucuronic acid. Okay, so when the glucuronic acid gets pulled off, we then create a new product called urobilinogen. Okay? And that's in the intestine. So now there's 3 places it can go. Number 1, number 2, number 3. So the first place it can go is back to the kidney. Okay, you can go back to the kidney to be, not the kidney, back to the liver. You can go back to the liver. Let's see, that one's actually number 3. This one's number 1. So number 1, you can go to the kidneys. And this is where you get yellow pee, because then it secretes it out. And that's called urobilin. Bilinogen. Urobilin. Okay. Number 3 goes to the liver, right, as I said before. So it can come back over here and it can get reconjugated again. Or the third thing it can do is it can stay in the intestines and this is what makes your poop brown. Pigment. And this is called stercobilin. So urobilinogen and stercobilin. Okay, and that's what it's called. So this is kind of how we break down red blood cells, and with a special emphasis on the heme part of the hemoglobin. Okay, so the thing to really remember is I feel like they're going to ask a lot of questions on this kind of a section with jaundice and those things. And so the thing to really understand with this is this part. And that is when you go from biliverdin to bilirubin, bilirubin is unconjugated and it comes into the bloodstream. If you have a problem with your liver, then what ends up happening is the bilirubin amount in the bloodstream increases. That can go to the cerebral cortex, or we'll just call it the brain. And it does something to the mitochondria. And it can be very deadly and dangerous if you have this high amounts of bilirubin within the brain, within the bloodstream. So sometimes oftentimes if you have high amounts of bilirubin, then it leads to an increase in jaundice, which is a yellow pigmentation. Sometimes you get it in the sclera of the eye or in the skin pigment. Nevertheless, jaundice does come. Okay? So we can get jaundice. And jaundice can come through here, but we can get jaundice if we have any blockage here as well. So we can have a blockage in the liver, and that means that we have unconjugated bilirubin. If we have blockage in the bile duct right here, if this was blocked, then we would have conjugated bilirubin when we would test it. And so we would know that the liver isn't the problem, but it's the bile duct that's the problem. And so we wouldn't have urobilin, and we wouldn't have stercobilin, and we wouldn't have, urobilinogen. We wouldn't see this glucuronic acid being taken off. So let me just name the important things again. You're going to break down the heme here. Heme is going to break down into iron, which iron will go to fibrinogen. Iron will go to ferritin. But then you're going to create bilirubin, or biliverdin. Biliverdin will then create, through an enzyme, bilirubin. Bilirubin is then unconjugated, and so it gets shoved into the bloodstream, and it's not water soluble. So then we take that bilirubin, and albumin secreted by the liver can come up and grab it and bring it into the liver. When it does that, it takes the unconjugated bilirubin and combines it with glucuronic acid. When glucuronic acid gets bound with this unconjugated bilirubin, we end up having a conformational shift that occurs, and we have conjugated bilirubin with glucuronic acid on it. And now it's water soluble because of the polarization of glucuronic acid. It then leaves, it goes to the bile duct. From the bile duct, we then take it into the duodenum through the sphincter of Oddi. Then in the duodenum, a couple things can happen. There's a bacteria in there called protease. Protease will then strip off the glucuronic acid. It will strip off the sugars, and we're left with a new molecule called urobilinogen. That urobilinogen can do three things. We can send it back. Number one, we can send it back to the liver to be unconjugated or conjugated again. Because when we take that glucuronic acid off, it becomes unconjugated again. So we can add glucuronic acid back on it and send it back through the bile duct. The second thing we can do is we can put it in, actually I need to make a note here really quick. Second thing we can do is keep it in the large intestine, or in the duodenum. And when we keep it in the large intestine, when we keep it in the intestines, in the duodenum, in the small intestines, then what ends up happening, when we put it in the duodenum, what ends up happening is that bacteria will cleave things off, like I already mentioned before. But if we keep it in the intestines, then that's what creates the color of the poop, which is stercobilin. And that's why the poop's brown. And we poop out that urobilinogen through stercobilin. Then we have urobilin, which is if number three, we send it to the kidneys, and the kidneys will turn it into that yellow pigmentation coloration in our urine, and we pee it out. And that's how we get rid of bilirubin and break down hemolysis, break down erythrocytes. One thing I want to mention over here was this word right here, and that is sometimes we don't send it to the liver. We can use blue light to cause this conformational shift and to cause it to be conjugated. And this is called a photoisomer. I believe that's what it's called. It's called a photoisomer. Yep. Okay, and that's everything for those sections. So I'm gonna scoot this puppy down here like that. Hopefully we can see all that there. Okay, and I'm gonna try to explain hemostasis pretty briefly, okay? So we've gone through blood, components, plasma, red blood cells, erythropoiesis, hemolysis, hemostasis, erythrolysis. Okay, so what ends up happening here is we have something called hemostasis. And hemo means blood, and stasis means to stop. And so really what we're talking about is hemostasis is clotting, stopping blood from leaving. So sometimes we can have an injury, or we can have, we can have this injury occur where we have a hemorrhage or a tear in the skin, and we bust the blood vessel. And so we have to have a way of clotting it or patching it. And so there's three ways we can do that. The first one is through vasoconstriction, but we call this a oh, what do we call this? We call this a vascular spasm. But I know that there's, vasoconstriction, so vascular spasm. Okay? And that is vasoconstriction. The second thing is platelet formation. Then, so we can have platelet plugs. Platelet plug formation. So we got platelet plug formation here. And that will use platelets. Then the third thing we can have, is a coagulation cascade. Okay? So, with the coagulation cascade and all these things, I'm going to draw out a cell here so we can kind of imagine kind of what's going on, okay? These are epithelial cells, or endothelial cells. Okay, and so what can happen is sometimes we can have a mechanism of injury occur. You can have a mechanism of injury. Cut or blood's leaking out, right? Can we see that there? So we got all this blood leaking out. So one thing that happens is the damaged endothelial cells can send out this muscular signal to the smooth muscle, and it sends out this signal. We don't need to know the name of it, but basically it binds onto a receptor on the smooth muscle, and it activates the smooth muscle to do vasoconstriction. Vasoconstriction to contract and decrease blood flow. So then we're not losing a ton of blood. So this is the first mechanism that we do. Okay? And this is called vascular spasm. Okay? The second thing that we do, okay, is in the same area, but just so I'm not drawing on top of everything. Well, I guess I could put it right here. We have this molecule, because over here there's these proteins here, and they have this negative charge in terms of polarization. And the negative charge attracts this thing. This is called VFW, okay, VFW. And it binds into this site here. And there's lots of them. They actually stand for something called the von Willebrand's factor. On the von Willebrand's factor, there is a receptor, glycoprotein 1B, GP1B receptor. One thing I failed to mention is within the cell, the bloodstream, we have these blue dashes which represent platelets. Okay, these are platelets. Okay, and what these platelets do is they're going to bind to the GP1B receptor on the VFW, Van Willebrand's factor. VWF. Van Willebrand's factor. Okay? And they're going to bind to that. So then let's draw in a platelet binding to that receptor. On here, there's two other receptors. There's a PYR12 or P2Y12, P2Y12 receptor. And what the P2Y12 receptor does is it takes in TXA2. And TXA2 will bind to that and activate platelets. So there's three ways platelets. The first one is TXA2. The second one is Van Willebrand factor. The third one is ADP. So we have another receptor here called ADP. And what ADP does is ADP activates these platelets for coagulation, or this sticking of them together, so, or platelet formation, right? So something can inhibit TXA2, and that's acids, NSAIDs, which would be our ibuprofen, and aspirin. Some other things can inhibit the formation of platelets, and something can inhibit actually the ADP, right? And what that puppy is well, let's see, P2Y12 actually goes with ADP, and TXA2 just kind of has its own receptor, but the P2Y12 actually goes with the ATP receptor. So this one's called Plavix, and Plavix inhibits the P2Y12 receptor. So then ADP can't bind, and then we don't have activation of the platelets, so that would decrease clotting. Okay? So those are some important factors that we need to know about this. And the last thing is that TXA2 is a type of fatty acid. Okay? So, again, Actually, I'll go over this at the end when it's fresh on my mind. But basically what's happening is we have a cut. We're gonna have this vascular spasm. We're gonna have muscle contraction going on. When muscle contraction happens, it's not gonna last forever. So we need to get something to plug this up. We do a platelet plug formation. This is called a platelet plug formation. And what happens is when these platelets bind, they actually expand and they get bigger and they build up in this whole area here, like this. They would build up on top of each other, and they would, if there was a cut here, you know, and this was the endothelial cell, and we would just build it up like this, and we would block any blood flow from being able to leave, and we just go around, right? Kind of like that. And so we would have this platelet plug forming on that membrane to block any blood from leaving. So we have the P2Y12 there. Something else that's really important for us to understand is this, and that is you have the three things that activate the platelets. You have the Van Waldebren factor, VWF. You have VWF, you have ADP, and then besides ADP, you have the TXA2 factor, which is a fatty acid. And so they're gonna either excite it, or if they're inhibited by the drugs, Blaxin, Plavix for ADP, and then NSAIDs, so like ibuprofen and aspirin for TXA2. Okay, so what ends up happening with this is something unique, and that is, this is one way to form clots, but there's two things that can happen that we don't. So this is called a thrombus. Okay, this is the thrombus, when we have this clotting building up. Now, what we don't want is for this thrombus to leave. We don't want it to float in the bloodstream here. Okay, we don't want this thrombus here. This would be called an embolus. So we need a way to anchor down this platelet plug formation. We do that through something called the coagulation cascade. And we create something called fibrin. And fibrin will be able to kind of plug that up for us. So, here we go. What ends up happening in this situation is fibrin, let's say we had our coagulation cascade going on here, our coagulation cascade assisting in this platelet plug formation. Okay, do this again, platelet plug formation. Platelet plug formation. So what ends up happening is, with this platelet plug formation, is we get something called factor 12. And that turns into something called factor 12A. And that uses the enzyme to then break down into factor 11 and factor 11A. And then we have factor 9 and factor 9A. And they combine with factor 8. And this is a cofactor, which assists in the enzymatic process. And factor 9, factor 8, then create something called factor 10 and factor 10A. And factor 10A will combine with factor 5. And together, they're going to make factor 2A. 2. Which then makes factor 2A. And we call this prothrombin. And and we call this thrombin. And then we have 2A breaks down into 1 and 1A. And this is fibrinogen. And this is fibrin. Fibrinogen and fibrin. And we are looking for fibrin, okay? And we call this the intrinsic pathway, okay? I'm trying to remember the drug that it's called that inhibits the intrinsic pathway. If I saw it, I would know it. So the intrinsic pathway is blocked by heparin. Heparin's a drug that blocks factor 12. And because it blocks factor 12, in order to test this, it's called a PTT test, okay? PTT test. Then we have something over here that comes from a cell on the outside. It's actually called a TF tissue factor, extrinsic pathway, but it's called the, yeah, so these are called extravascular cells, the extravascular cells, right? Okay. So the extravascular cells, what they do is they create something called the extrinsic pathway. And the extrinsic pathway is extrinsic because it's coming from out of the cell. But what this does to create fibrin, because that's the end goal, is to create fibrin, which acts as this net mesh to go around this and hold it down in place. So the extrinsic pathway actually starts with something called the TF factor. I believe that's what it's called. Tissue factor 3, yeah. So it's called tissue factor 3. And what ends up happening is tissue factor 3 will lead into something called factor 7. And that turns into, through an enzymatic process, to factor 7A. And then factor 7A will go into factor 10. Yeah, and 10A. Then 10A will combine with 5, and we go back up to here. So this process here then takes us back up to here. And so we call this the common pathway. Okay? Then that's called the common pathway. And then it can create fibrin that way. And so there's drugs that we can use to block this. So if someone had a heart attack or they have clotting going on and we want to give them a blood thinner, we can use Calmid. I know it starts with a C. It's called calmodin. It sounds like calmodulin. Calmodin. We can use calmodin. Okay, and we can also use warfarin. And what they do is they block factor 7. Okay, they block factor 7. And when they block factor 7, you can no longer clot that way. And we decrease clotting, which decreases viscosity of the blood. Okay? Let me make sure I got everything here. Oh, and it does that because it destroys, let's just put inhibits vitamin K. So it destroys or inhibits vitamin K, which warfarin, calmodin, they do that. And factor 7 needs vitamin K. But if you inhibit it, then you inhibit the factor 7. And this uses a PT test. And it uses IPM, INR for the results. Okay? So that's about everything that we need to know for this unit, or this section. So last thing I'll do is I'll kind of go over the hemostasis one more time. In hemostasis, you have blood vessels that have now ruptured due to a hemorrhage or an injury. In this case, you have blood spilling everywhere. In order to clot or patch that, your bloodstream will do that naturally in 3 ways. The first one is it's going to cause a vascular spasm. The injury to the endothelial cells actually causes like nociceptor reaction. It causes, in a way, we don't need to know the name of it, but it's going to activate the smooth muscles. When it activates the smooth muscles, it actually is going to tell the smooth muscles, hey, we need to do contraction. So we're going to do vasoconstriction, and we're going to decrease blood flow. When we decrease the blood flow, we're in a good stage now because now we have less blood leaving because we have less blood coming, okay? The second thing that our body does, second mechanism, is we're going to do a platelet plug formation. And the way that it does this is in that injured area, you have proteins within the outside of the cell that are negatively charged. When those negatively charged ions are exposed, they pull and attract something called a Van Waterbrand's factor. We just call it VWF. This binds in that hole or that section where the blood's leaking out of. On it, it has a receptor called a glycoprotein 1B receptor, GP1B. That attracts and activates platelets. When platelets are activated, they expand. And then they actually have something in between them, which I forgot to explain, but the platelets, if we bound to another one, this is called fibrinogen. Okay? Fibrinogen will connect the platelets together. And then these are called like G, B, P, 2A, 2B, and 3A, right? Let me see what they're called exactly. Yeah, GP, 2B, and GP, 3A. Yeah, I got those. But that's what's going to connect those platelets together, okay? There are 3 things that activate them. I already mentioned the first one, VWF, Van Waterbrand's factor, activates them. ADP activates them through a receptor on the platelets called, they're activated on the platelet through, the platelet gets activated through this receptor. It's called P2Y12, and that's what ADP binds to then turn on the platelet to start accumulating in an area and start expanding. Then the third one we have is the third one that we have is going to be TXA2. And this is a fatty acid, and what TXA2 does is it can also activate the platelet. Now, let's talk about some drugs that inhibit those two things. Let's start with TXA2. TXA2 is inhibited by NSAIDs drugs, so ibuprofen and aspirin inhibit that binding site, so then we don't turn on as many platelets, so we have less clotting. With the P2Y12 receptor, that belongs to the it belongs to the ADP binding site. That one gets inhibited by a drug called Plavix. I knew it started with a P. Plavix. And what Plavix does is it blocks that, so then inhibits that, so then we have less platelets being activated, which is less clotting, which is not good in terms of a bleeding injury. So that's how we turn on, turn off, well, not turn on, but that's how we control platelet activity for the platelet clot, platelet plug, platelet plug formation. Okay, the third thing is the coagulation cascade. And this is going to ultimately make fibrin, and fibrin will act as a mesh net to hold that platelet plug formation in place, so we don't get an embolism forming, right? So I talked about having a thrombus, which is that clumping to plug the spot, and then we don't want it to leave and float into the blood and cause a blood clot. So we have something called fibrin that will act as a fibrous net and keep it in place there, creating the thrombus, but we don't want an embolus to occur, which is that floating away. So what ends up happening is, for the coagulation cascade, is you have factor 12, and this is for the intrinsic pathway. You have factor 12 goes to factor 12A, which goes to factor 11A, which then goes to factor 9. 9A combines with 8 cofactor, which create factor 10, which create factor 10A, which combines with factor 5, and they together will create factor 2. And then, right, so they come together and they create factor 2, which is called prothrombin. Then they create factor 2A, which is thrombin. Then factor 2A thrombin will create factor 1, which is fibrinogen. And then factor 1 will create factor 1A, which will be our fibrin, which is what we want. Now, we're plugging it up. So let's talk really quick about ways that we inhibit that pathway, the intrinsic pathway. We use something called heparin, which blocks factor 12 and inhibits coagulation and clotting. Could be dangerous. Okay, so that blocks intrinsic pathway. Then we have extrinsic pathway. Difference is we start with TF factor 3, so tissue factor 3 in the external vascular cells. So on the outside extravascular cells, and TF3 will then activate factor 7, and factor 7 will then turn to factor 7A, and then factor 7A turns to factor 10, 10 goes to factor 10A, and so we create this cascade again, and now we're back into the common pathway. So 10 down to fibrin 1 is going to be the coagulation cascade common pathway. But what we're focusing on as well is what drug is going to inhibit the extrinsic pathway. And calmodin does that, and warfarin. And they do that by destroying vitamin C, or vitamin K, which vitamin K is a huge cofactor, I would assume, or helper for factor 7. And if we destroy vitamin K, then factor 7 is no longer able to do its thing, and clotting goes down. And we inhibit that extrinsic pathway. I don't think there's anything else. So I think that's everything. Awesome.